As chair of the Federal Committee of the Plant Conservation Alliance, I am pleased to introduce Dr. Tanisha Williams, postdoctoral fellow in botany at Bucknell University. Tanisha is the Richard E. and Yvonne Smith postdoctoral fellow in botany at Bucknell University, and her dissertation research examined the impacts of climate change on species throughout South Africa. Her postdoctoral research explores the role of Aboriginal peoples, or the role Aboriginal peoples have on the movement and maintenance of plant species, and understanding how biogeographic barriers impact species distributions in Australia. She also uses genomics methods to update the conservation status of rare plants and has extensive science communication and policy experience, which is very important in our jobs, and is the founder of Black Botanists Week, a campaign to amplify diverse voices in botany. Tanisha completed her PhD in ecology and evolutionary biology at the University of Connecticut in 2019. And so I'm happy and hoping that you all will join me in welcoming Dr. Tanisha Williams, who will talk to us today about protecting rare and endangered plants through university and governmental partnerships.
Thank you so much, Patricia, for that wonderful introduction. I just wanted to know, what slides are you seeing, and then can you see me or hear me?
Both. See, hear you, and we can see your front slide. Thank you.
Okay, great. I is so excited to be here. I see some familiar faces. So hello to everyone that I know, and hello to everyone that I don't know, and hopefully we'll become fast friends after this talk. So I'm excited to be presenting about this amazing partnership that I like to call Boots on the Ground, Boots in the Lab, protecting rare and endangered plant species through a university and governmental partnership. And so this work is done in collaboration with Dr. Chris Martin, whose lab I am postdocing in, our honor student, John Hayes, PA Natural Heritage Program, Rachel Gold, who's a botanist there, and moss and botanist expert, Dr. Scott Schutte from the Western Pennsylvania Conservancy. So when you think about Pennsylvania, you may not think about a state overflowing with biodiversity. I'll speak for myself. When I thought about Pennsylvania, that's not something, biodiversity wasn't high on my list. But due to the geological history, it has some very interesting landscapes that have given rise to phenomenal biodiversity throughout the state. So there are also a significant number of rare species throughout Pennsylvania. And it's associated with substrates such as serpentine, limestone, and peat. PA is separated into 7 major ecosystems that I'm going to go through really quickly. So we have the forest ecosystems that are a major ecosystem throughout Pennsylvania. By the end of the 19th century, nearly all forest land had been cleared or burned. But today, Pennsylvania has about 17 million acres of forest that cover nearly 60% of the state. And almost all of them are classified as second growth forests, but we do have some old growth forests as well. And so it's a wonderful state if you love hiking and getting outside like I do. Grasslands make up about 26% of the land cover, and we only have a few true prairies intact. Barrens such as shell and scrub oak barrens are rare in the state. They comprise about 3% of the land area. And we have quite a bit of subterranean areas such as caves and these specialized habitats for bats and other small mammals. And most of the animals that are associated with these areas and these ecosystems are often listed as threatened and endangered. More than 50% of Pennsylvania's wetlands have been lost, and so we have only about 400,000 acres left in PA. PA has an impressive 83,000 miles of streams, and this is second only to Alaska in the number of stream miles. So it's a wonderful place. If you like hiking, if you love kayaking, canoeing, it's wonderful. Actually, where I'm sitting right now, the university is right off the Susquehanna River. So many of the stream miles have been negatively impacted by industry, and along with these large number of stream miles, we have an oppressively large number of waterfalls that I'm showing here. So there's a state park called Ricketts Glen State Park that's close to me. And in that state park alone, there's 22 waterfalls, and they're just as amazing as you can imagine. So it's a wonderful place to live and a wonderful place to work. We have suburban and urban communities. That red star, I'm in Center County right now, so I'm showing you I'm smack dab in the middle of the state in rural Pennsylvania. And so these suburban and urban communities account for about 15% of the land cover, but that's rapidly increasing, as we know, as we are spreading out and out into the communities. Development has increased, and so this is just an old statistic from 1982 to 1997. The development had increased from 100 acres per day to 300 acres per day, and so we are seeing this trend continuing to increase. So all of these ecosystems that I just went through very quickly give rise to the almost 7,000 native species that are found throughout the state, and there are over 3,000 plant species. And so I want to jump right into the plant species. So we have about 3,400 plant species throughout Pennsylvania. 2,300 of those are native and naturalized, but Pennsylvania's wonderful biodiversity is under threat. So out of those 2,300, we have about 600 of the native plant species are recognized by PADCNR, Department of Conservation and Natural Resources, to be considered rare, threatened, or endangered. The threats to Pennsylvania biodiversity are our usual suspects, as I like to call them. It's climate change, habitat loss due to development, invasive species, really we're talking about encroachment here, and selective browsing by white-tailed deer, especially in this region. So the species that I'm going to talk about today really are being impacted, number one, by habitat loss, number two, by invasive species, and then thirdly, by climate change. So how do we go from losing to winning? How do we conserve our native biodiversity here in Pennsylvania? So we get a team of governmental scientists, so Rachel from the Natural Heritage Program and Dr. Scott Schutte from the Western Pennsylvania Conservancy. And these are what I like to call the boots on the ground. And they team up with university faculty, postdoc, graduate students, undergraduate students, who I like to call boots in the lab. And so we combine our powers together to help conserve native biodiversity and update the conservation status of rare plants throughout the state. This partnership has been done on another plant species, and we're continuing to work together, but I'm just going to share a few of our most recent plant species. We've done work with Erigenia bulbosa, also known as the harbinger of spring, and I'm wishing for some warmer weather soon-ish, not too soon, but soon. So Erigenia is endemic to the eastern United States. It's one of the earliest blooming wildflowers in eastern North America. And in Pennsylvania, it exhibits this east-west disjunction distribution. So the little map that I'm showing there, when I brought out the legend of that map, is that current populations are circled in black, and those are populations from 1993 and later. And we could see some of those populations, historic populations that are extinct or no longer with us, or this is from 1992 earlier, and we could see some of those populations throughout the state. The map that's underneath that, the larger map, is showing this east-west disjunction and the populations that were sampled for this paper here done by the previous postdoc in this position, Dr. Angela McDonald, and also Cheyenne Moore, who's on the call right now. So the reason why we were interested in this species is that the species was of conservation concern because the Pennsylvania Department of Conservation and Natural Resources, I'll start saying PADCNR for now on, had an environmental review process, and it deemed that the population in the eastern part of the state, so here we're seeing those population that's Safe Harbor and York Furnace, those yellow and green populations are subject to regulations, while population in the west part of the state, so those reds and oranges and browns, we're seeing Cedar Creek there and Ryerson Station State Park right there, were not under any regulations. And so based on the work that was done by Dr. Angela McDonald and Cheyenne Moore, their work showed that all the populations were genetically unique. It was thought that those western populations near Philly were going to be one unit clustering together, but as we can see here in the structure analysis, all of those populations are genetically distinct. So all populations also exhibited low heterozygosity and high levels of inbreeding, and these are all no-nos that we don't want for rare species, that we don't want for endangered species or any plant populations. And so this work that was done by the lab recommended to DCNR that conservation and regulations should be applied evenly throughout the state. And the conservation status of the species has been updated by the Western Pennsylvania Conservancy, the Natural Heritage Program, and also at the state level. And it's now classified as critically imperiled across the state evenly. We've also done work with Baptisia australis, and this work was done by Cheyenne Moore. It was her master's thesis work. And I'm going to actually let Cheyenne introduce this work herself through a video.
How can a perennial wildflower that many of us have in our gardens be considered rare? And what do massive blocks of river ice have to do with it? We're going to find out on the next Plants Are Cool Too. Animals are cuddly, animals are cute. You could put a kitten in a three-piece suit. But could an animal make its own food? Could an animal feed the whole world? Could an animal help you get a girl? Plants are cool too. Plants are cool too. Plants are cool too. It's an early, misty morning in western Pennsylvania as we head out on the Allegheny River, a 325-mile river course whose waters go on to join the Ohio River and, eventually, the mighty Mississippi. Somewhere downstream, we intend to find graduate student Cheyenne Moore, along with their collaborator, Scott Schutte, who's out here looking to learn more about one of the coolest imperiled plants in the state and the unusual riverside habitat it grows in. A research project stemming from her lifelong love of science and green things.
I always knew I wanted to be a scientist, but I had a position working outside in college, and I really enjoyed it. And I wanted to be outside, and I wanted to learn about the plants because plants tell us a lot of cool things and are often overlooked. And so that was really important to me, to look at something that was really cool in the environment that people don't really pay attention to, but we can learn a lot from.
It wasn't long before my son Jackson and I tracked down their kayak, pulled up on a gravelly island, one of about a half dozen similar habitats Cheyenne was planning to survey on that day. We'd learned soon enough that these particular formations are called scour prairies, and they harbor some pretty fantastic plants. Hey, Cheyenne, good to catch up with you.
Hey.
How are you?
Good, how are you?
Awesome. So you seem to have found some pretty cool plants.
I have.
Yeah, what are you doing out here?
So I'm looking at this plant, Baptisia australis, or blue false indigo.
Okay.
Which is a state threatened species.
Yeah.
And so I'm out here seeing where it is along the Allegheny River.
People might.
Oh, I'm not sure what happened. Only a few more seconds of this. I just want Cheyenne to
actually have this plant in their garden. So why are we calling it rare out here in the wild?
So in Pennsylvania, it's considered state threatened because in Pennsylvania, it's only really found along riverways. Because it's in these communities called scour prairies, which are facilitated by ice scour along waterways, where anywhere else you might find it in a prairie in the Midwest or something like that.
So it's more common to the west of us, but what are we here, like on the eastern edge of its range?
We're on the northeastern edge of the species range.
The species only occurs in these unusual scour prairie habitats.
Essentially, yeah, it's only along 4 waterways.
All right, so tell me about this. How does ice scour work? Where's it come from?
Whenever ice forms in the wintertime along these big rivers, it eventually breaks up, and those big sheets of ice that are broken up come along the riverbanks and basically scrape the banks enough that it reduces trees and things that might shade these individuals out, and it allows them to continue growing with other prairie species like big bluestem and Indian grass.
All right, I'm going to stop it there, but I'm going to.
So it could be.
Or maybe not. It's not going to stop there. Okay.
How can a perennial wildflower that may...
Okay. Oops, going too fast. Let's see. So I want to highly encourage you all to go to the YouTube channel, Plants Are Cool Too. This is done by Dr. Chris Martin, and I left that video on a cliffhanger. So if you want to know more about Baptisia australis, please look at that video, and Cheyenne will tell you all about what's happening with Baptisia australis throughout Pennsylvania. So our latest project that we're working on is Casmantium latifolium. So I worked on this project with undergraduate researcher and our honors student, John Hayes. So Casmantium latifolium is commonly known as river oats or wild oats. The name is fitting because this species loves riparian habitats, and it's found along many riverbanks. It's part of the Poaceae grass family. It's a really nice looking grass, and I don't say that often, although I've also worked with switchgrass for a second of my scientific career, and I thought that was a nice looking, beautiful grass as well. It's perennial, it's characterized by these large, flat, drooping spikelets that are often called fish on a string. It's popular in cultivation as well because it's such a nice looking grass. And that's me showing in one of the ornamental plantings at the University of Georgia. It has many ecological significance, such as it prevents soil erosion, improves water quality, is one of the host species of this pepper and salt skipper. It's a minor food source for birds and rodents, coverage for insects, and also the stems and leaves are used as nesting materials for certain birds as well. So it's important to its ecosystem. Casmantium latifolium is distributed throughout the southern Midwest and eastern United States, and it also goes into Mexico. Throughout its range, it's considered globally secure. However, in Pennsylvania, the Pennsylvania Natural Heritage Program classified this species as critically imperiled, yet at the state level, Casmantium was tentatively undetermined at the time that we were doing this work. So the Pennsylvania Natural Heritage Program gave the ranking of critically imperiled because PA represents the northeastern edge of its range and its distribution. There are 3 disjunct populations. So I'm showing here on this map the Monongahela river system in the west in that magenta color, the Juniata River system, which is a central population with the blue star, and the Susquehanna, which is the eastern population, highlighted in orange here. And the populations have been noted as declining because habitat elimination due to agriculture, damming of rivers, or altering flood patterns, which are impacting the species that loves riverbanks, loves riparian areas, and also competition from invasive species. So these were the criteria and why the Natural Heritage Program was concerned about this grass species and native species to Pennsylvania. So our team, we use updated genomics methods to assess the genetic diversity, levels of inbreeding, and population structure of the remaining populations of Casmantium latifolium throughout Pennsylvania. And so our ultimate goal was to make sure that we were updating the conservation status at the state level and with the Natural Heritage Program if needed. So collections were done of all 11 extant populations, so the last populations remaining in the state. with collections ranging from 7 to 16 individuals per populations, totaling 155 population. My name is there because I did go to the one population, Ernie Creek, and it's nothing, I always tell my student, it's nothing like seeing your plants in their native habitat, but I have to give all the credit to Scott and Rachel that went around the state collecting this species. All DNA extractions, quantification, and quality checks were done by John Hayes, Ariel Anton, who is another undergraduate student. She's pictured there next to John. John's in the orange shirt, Ariel's on the left in the greenhouse. And then also with help from Cheyenne Moore, she's in the middle helping John with the extractions. Sequencing assembly was done using iPirad, and all PopGen analysis were done in R. And so this pipeline was initiated and established in the lab by the previous postdoc, Dr. Angela McDonald. I further optimized the pipeline after going to this class in the Czech Republic called the Workshop on Population and Speciation Genomics. It's a wonderful, wonderful class, and I think there's some similar classes also here in the US. But this really helped kind of jumpstart my genomics data analysis skills. So we're going to hop right into the results. So we ended up with a data set of 133 individuals across 11 populations. We had about 2 million reads per sample on average, tons of high quality data. We were super excited about this. And our ending data set had 1,000 loci across almost 2,000 SNPs. So we were ready to jump right into our genomics and our population genetics analyses. So the first thing we wanted to look at is gene flow parameters. So we looked at an overall FST across all of those 130 individuals, and our FST value is just 0.1130. So the FST scale goes from 0 to 1. The closer you are to 0, you have low genetic differentiation. Once you get above 0.15, this is moderate to significant levels of genetic differentiation. So we can see that we have kind of low genetic differentiation between these species right now. So we wanted to delve in a little bit further. And we looked at a pairwise comparison, and this really makes the picture very clear. So the redder you are on the heat map, the more genetically distant these populations are from one another. And so I'm grouping these populations based on the waterways. So we can see in those Western populations here, and I'm circling those populations on the map, there are a lot of gene flow in between those populations. That lighter color is saying that you really can't tell these populations from one another. So there's a lot of gene flow that's happening. Then we look at that there's really very little gene flow between Western and Central or Eastern populations, right? When you're doing that pairwise comparison, those squares are very red. And then the next, there's very little gene flow between Central and Eastern populations. And so all of those red colors are above that 0.15 threshold. Next, we looked at the levels of genetic diversity overall and within each of the population. And this was such a good sign that we heard. So HO is our observed heterozygosity, this measure of genetic diversity, and HE is expected. And in every case overall and within each population, the observed heterozygosity was greater than the expected. So that's a great thing because normally with rare and edge-range species, you see the opposite. We also found that there was no inbreeding within these. So the inbreeding scale goes from a +1, which positive numbers signify inbreeding is occurring, and negative numbers signify that inbreeding, there's outbreeding happening, and you don't have this signature of inbreeding. So overall, we see a negative values, and then when we look at the FIS, that inbreeding coefficient across each of those populations, we see there's no signature of inbreeding. And also our confidence interval, so that 95% confidence interval doesn't overlap 0, so we're confident that these values are true. And so that's a good sign. We don't see any inbreeding depression signatures within these populations. We conducted a structure analysis, and the best ancestry estimation was K equal to 5, so 5 ancestral lineages. There's a great deal of admixture within these populations. So reading this graph from left to right, we can see that those eastern populations really break it up into 3 units, kind of that orange, gray, and then red units. The central population is clustering together to the closest eastern population, that's Ernie Creek. So we're seeing that red between Ernie Creek and also the Juniata central population. And then that western population, all of those clustered together under that green color. They're one genetic unit. So what we're really seeing here is this east-west disjunction that I kind of mentioned at the beginning of this section. So we're seeing the central population is clustering with the east, and that western population is clustering as this genetic unit separate from all other populations. And I should also mention that the central population, that Juniata, is a tributary of the Susquehanna, that those eastern populations. So let's review these results. So we saw that all populations were genetically healthy. There were no inbreeding and high genetic diversity within these populations. So Western populations seem to be genetically similar and secure, having high levels of gene flow between these populations. While on the flip side, Eastern and Central populations were genetically isolated from one another, and they were also isolated from the Western populations. So we see that there's a potential for inbreeding within these populations because they're so isolated from one another, and they are at greater risk for things like genetic drift, and so are of greater genetic concern. So in conclusion, we know that state populations are limited and declining due to habitat loss, like I said, from damming and industry and development and things like that. Invasive species are a problem at some of these riverbank sites. But we are excited to say that the remaining populations do seem to be healthy, and in most instances, we do see massive population counts. So we can see Rachel there in the background at one of the sites, and all along the ground, that's all Casmantium latifolium. And I will say that Ernie Creek site was on private land, and there was a farmer who graciously let us come onto his property and talk to us about different things that were going on. But along the river that was going through his property, Casmantium latifolium was just all over the understory. And it was just impressive to see the population so massive like that. And we are seeing that Western populations are appear to be genetically secure, like I've said, and those Eastern and Central populations, we really need to assess those as well, but they're not looking as healthy and as great as the Western population. So in our next steps, our work is actually in review right now. We're waiting to hear back from a journal about the fate of our paper. So we're excited about that. And this is John's first author paper. So we're really excited about that. This work has been presented at the Pennsylvania Rare Plant Forum and at the Botanical Society of America's Botany Conference. We presented this work twice, once when we first started this, and then another time once we have results and other conclusions. So here is a slide of us giving John the thumbs up that he passed his honors thesis, presenting on some of this work. And because of all the work that has been done on Casmantium latifolium, we have updated the status across the state. And we kept that same status that the Pennsylvania Natural Heritage Program, that S1 class of critically imperiled, and mainly because there is no connection between that east-west disjunction. We have no connection between the Western, Central, and Eastern populations. And so we need to save all of those unique genetic units because we only have 11 populations left throughout the state. I have to thank all of our funding sources. This was a team effort by so many folks to help us get this project up and running, and all the data collection analysis and things like that. I would also like to thank the many scientists who have made this project and all of the projects that I've mentioned possible. This has been a really fruitful partnership and something that I've really come to enjoy while being here at Bucknell University. I'm originally from Washington, D.C., so I always say that policy runs in my veins, and so it's really nice to partner up with governmental scientists and agencies, botanists and scientists to do this work and to know that we're changing policies, we're changing laws, we're actually conserving these species that we care so deeply about. So it's been such an amazing experience for myself as a university scientist working with folks outside of the university. Some updated news that I have to share. We got another grant to work on some more species. We're actually working on, I think 4 species, right? 4 Pennsylvania rare, threatened or endangered species here in the lab. And this is one of the projects that I'm heading, this population genomics and climate change vulnerability of Appalachian endemic plant species in Pennsylvania. So we're working on bog Jacob's ladder. I'm super excited about this project because not only are we doing the population genomics of this work, and we're including a range-wide study of this work to make a really scientifically sound and informed recovery plan for this species. We only have about 7 sites within Pennsylvania that are left of Jacob's ladder, or the bog Jacob's ladder. And within those sites, we have sites that only have as low as 3 individuals upwards to 300. And the norm is closer to the 3 sites. So we're really concerned about this species. And within the state, it is an endangered species across its range. It's globally at risk. And so we're excited to take on this project this summer. I'll be working with Cheyenne, I'll be working with Rachel, I'll be working with Scott. And I also have a undergraduate student here at Bucknell, Andy Dorsal, who'll be helping with this project as well. So I'm hoping that he'll take this on as an honors thesis to do similar work like John. And I'm excited that we're adding in the climate change aspect to this. So we're doing the genomics, climate change, we're doing habitat assessment of this species using NatureServe protocols. We're really giving this species the workup, and I'm excited to conserve this species as well. So something that's not about native plants here in the US, but native plants across the way in Australia, I just want to highlight some of the other work that we're doing in the lab. We just had a new species description that was out of Solanum scalarium. It's a bush tomato in the Northern Territory of Australia. This is this bush tomato, as we say, was hiding in plain sight. It's actually found in a national park near a trail that's heavily walked. And Dr. Chris Martin, who's pictured here throughout this slide, noticed these prickles along the male rachis. And he was like, man, this is just something pretty different. I haven't seen this before. And luckily they had the permits and they were working with Australian botanists and herbarium curators. They took some samples. Some of the plants had seeds, brought the seeds back here to Bucknell University. We grew them up, and we had an undergraduate student who is also helped. So John Hayes and some other folks that helped to grow this plant up, measure this plant, and scalarium is the Latin name for ladder. And there's 3 kind of reasons why we named it scalarium. One, that key characteristic that Chris found out, those prickles, as we can see the male, these prickles right here that we're picturing. This is something unusual for this group. So that ladder-like prickles also was found near some steps, so ladder steps. And then lastly, we wanted to highlight this access to nature, who feels safe in nature, who doesn't feel safe in nature, who you see on hiking trails. And I normally don't see anyone that looks like me. And so we just kind of highlighted some of these things within the article as well. We've been getting a lot of press on this article, done some local news, as well as There's a new episode on NPR's shortwave podcast where I talk to the folks at NPR about this work and about the naming and about just making sure we say this is a new species description. We've uncovered some things new to Western science, but we didn't discover anything. We know Aboriginal peoples know what this plant is, and we do highlight that. This work is done, all of this work, Native Plant Societies, the Australia work that we're working on, and many of the other projects that I don't have time to go into right now that we're working on are done by a host of undergraduate students. I'm at a primarily undergraduate institution, and the students here are phenomenal. We go up to master's level, so Cheyenne was a master's student. We have a new master's student that's working on Pachystoma right now. And so there's so many people involved, so many folks. Our postdocs, Dr. Angela McDonald, Dr. Jason Cantley was a previous postdoc of the Martine Lab, are still heavily involved in these projects. And Dr. Peter Jobson is actually, he helped with that Solanum project, the discovering of, species description of Solanum scalarium, and he's associated with the herbarium in Darwin. And so I'm just going to talk about one last thing. I'm going to say two last things very quickly. So as Patricia mentioned, I started Black Botanist Week. It's something that's near and dear to my heart. This was an online initiative started in 2020 to amplify and find more Black people who love plants. With this organization, there are other folks that I'll bring up in a second. We've redefined the word botany and who classifies as a botanist. My saying is, and what I try to promote during this week and throughout the year, is that if you love your plants, you're a botanist. That's all you need, is a love for plants. So please visit our website, engage with us. We've put together on the website, and I manage the website, we put together a nice infographic that kind of tells about our reach for the first 2 years of the program. We also have a nice and impactful promotional video that describes why we all started this work. So lastly, I want to highlight the Black Botanist Week committee members. I can't highlight them enough. I cannot have done this. I cannot continue to do this work alone. These people, as I always say, they showed up and they continue to show up and they continue to do the work. So please amplify their work and invite them to give scientific presentations at your institutions. And we like to say the committee is diverse as the plants that we love. We're from the US, we're from South Africa, and we're from the UK. We are school teachers, we are postdocs, we are faculty members, conservation managers, botanical illustrators, and photographers. We are diverse as the plants that we love. So again, I want to thank this wonderful team of Black botanists for inspiring me. And I hope they will inspire you as well as you get to interact with them. So I want to tell you that these are all of our logos from 2020 up until 2022. I want to tell you all to be on the lookout for Black Botany Week 2023. I have no idea when the date is going to be, but we like to stick around to July and August. So the reason why last year's was a little bit later than our normal year, we actually, some of us went to the Botanical Society of America's, the Botany Conference in Alaska. And we actually held a symposium entitled The Black Botanical Legacy. And it was just so wonderful to actually meet everyone in person. I worked in South Africa as a graduate student, and so I knew our South African members, or 2 of our 3 South African members, and I've actually physically met them in person pre-pandemic, but everyone else on this committee I have not met in person. I've only been doing things online. So to meet some of these folks in person, that it was just a wonderful experience. And so just be on the lookout for our week that's coming up in this year. And something that I think would be really interesting to this group, the Applications in Plant Sciences Journal has a special issue on conservation. So the title or the theme is From Theory to Practice, New Innovations in Their Application in Conservation Biology. They want best practices, novel conceptual frameworks, software or databases, best practices for managing in situ collections, new techniques to combat poaching, et cetera, et cetera. And so abstracts up to 300 words are due on April 4th, 2023, and our group are definitely submitting an abstract to this special issue. And I hope that you all on this call, all of the wonderful things that you're doing, I hope you all submit something to this special issue as well. And so with that, I just want to thank you so much for your time. Thank you for inviting me. I'm super excited to hear more about the work that you're doing, and I'll hand it over and have time for some questions.
Great, thank you so much. I can't turn my camera back on and clap at the same time, but there are a lot of applauses showing up there. And yeah, that was really fabulous to hear your presentation. Thank you. We're going to open up for questions